A peroxide is a compound containing an oxygen-oxygen single bond or the peroxide anion, O2-2. The O-O group is called the peroxide group or peroxo group. In contrast to oxide ions, the oxygen atoms in the peroxide ion have an oxidation state of -1. The simplest stable peroxide is hydrogen peroxide. Superoxides, dioxygenols, ozones and ozonides are considered separately. Peroxide compounds can be roughly classified into organic and inorganic, whereas the inorganic peroxides have an ionic, salt-like character. The organic peroxides are dominated by the covalent bonds. The oxygen-oxygen chemical bond of peroxide is unstable and easily split into reactive radicals via homolytic cleavage. For this reason, peroxides are found in nature only in small quantities, in water, atmosphere, plants, and animals. Peroxide ion formation has recently been highlighted as one of the main mechanisms by which oxides accommodate excess oxygen in ionic crystals and may have a large impact on a range of industrial applications including solid oxide fuel cells. Peroxides have a bleaching effect on organic substances and therefore are added to some detergents and hair colorants. Other large-scale applications include medicine and chemical industry, where peroxides are used in various synthesis reactions or occur as intermediate products. With an annual production of over 2 million tons, hydrogen peroxide is the most economically important peroxide. Many peroxides are unstable and hazardous substances. They cannot be stored and therefore are synthesized in situ and used immediately. In biochemistry, peroxides are usually very reactive and thus occur in nature only in a few forms. These include, in addition to hydrogen peroxide, a few vegetable products such as ascaridole and a peroxide derivative of prostaglandin. Hydrogen peroxide occurs in surface water, groundwater and in the atmosphere. It forms upon illumination or natural catalytic action by substances containing in water. Seawater contains 0.5 to 14 ug/L of hydrogen peroxide, fresh water 1 to 30 ug/L and air 0.1 to 1 parts per billion. Hydrogen peroxide is formed in human and animal organisms as a short-lived product in biochemical processes and is toxic to cells. The toxicity is due to oxidation of proteins, membrane lipids and DNA by the peroxide ions. The class of biological enzymes called SOD is developed in nearly all living cells as an important antioxidant agent. They promote the disproportionation of superoxide into oxygen and hydrogen peroxide, which is then rapidly decomposed by the enzyme catalase to oxygen and water. Formation of hydrogen peroxide by superoxide dismutase peroxisomes or organelles found in virtually all eukaryotic cells. They are involved in the catabolism of very long-chain fatty acids, branched-chain fatty acids, diamino acids, polyamines, and biosynthesis of plasmalogens, ether phospholipids critical for the normal function of mammalian brains and lungs. Upon oxidation, they produce hydrogen peroxide in the following process. FAD equals flavin adenine dinucleotide catalase. Another peroxisomal enzyme uses this H2O2 to oxidize other substrates, including phenols, formic acid, formaldehyde, and alcohol, by means of the peroxidation reaction, thus eliminating the poisonous hydrogen peroxide in the process. This reaction is important in liver and kidney cells, where the peroxisomes neutralize various toxic substances that enter the blood. Some of the ethanol humans drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2 accumulates in the cell, catalase converts it to H2O through this reaction. Another origin of hydrogen peroxide is the degradation of adenosine monophosphate, which yields hypoxanthina. Hypoxanthina is then oxidatively catabolized first to xanthina and then to uric acid, and the reaction is catalyzed by the enzyme xanthina oxidase. The degradation of guanosine monophosphate yields xanthine as an intermediate product, which is then converted in the same way to uric acid with the formation of hydrogen peroxide. Eggs of sea urchin, shortly after fertilization by a sperm, produce hydrogen peroxide. It is then quickly dissociated to O radicals. The radicals serve as initiator of radical polymerization, which surrounds the eggs with a protective layer of polymer. The bombardier beetle has a device which allows it to shoot corrosive and foul-smelling bubbles at its enemies. The beetle produces and stores hydroquinone and hydrogen peroxide in two separate reservoirs in the rear tip of its abdomen. When threatened, the beetle contracts muscles that force the two reactants through valve tubes into a mixing chamber containing water and a mixture of catalytic enzymes. When combined, the reactants undergo a violent exothermic chemical reaction, raising the temperature to near the boiling point of water. The boiling, foul-smelling liquid partially becomes a gas and is expelled through an outlet valve with a loud popping sound. Hydrogen peroxide is a signaling molecule of plant defense against pathogens. In firefly, oxidation of luciferins, which is catalyzed by luciferases, yields a peroxy compound 1,2-dioxetane. The dioxetane is unstable and decays spontaneously to carbon dioxide and excited ketones, which release excess energy by emitting light. Hydrogen peroxide. The most widely used synthesis method of hydrogen peroxide is the Anthraquinone process. There, anthraquinone is first hydrogenated to anthrohydroquinone. This reduced compound is oxidized with molecular oxygen, regenerating anthraquinone and releasing hydrogen peroxide. Direct synthesis of hydrogen peroxide from hydrogen and oxygen is inefficient and currently is not practiced industrially. Many peroxides of mineral acids, such as peroxodisulfates and percarbonates, can be obtained by anodic oxidation of the respective acids. The anode material must be stable to the required high potentials of a few volts and therefore is either platinum or its alloys. Peroxydisulfuric acid was historically used for the production of hydrogen peroxide in a method developed in the early 20th century. This process requires relatively high concentration of peroxydisulfuric acid as its more dilute solutions evolve oxygen gas instead of peroxide. Inorganic peroxides. Inorganic peroxides are divided into ionic and covalent peroxide. The first class mostly contains the peroxides of the alkali and alkaline earth metals, whereas the covalent peroxides are represented by such compounds as hydrogen peroxide and peroxymonosulfuric acid. In contrast to the purely ionic character of alkali metal peroxides, peroxides of transition metals have a more covalent character. It should be noted that in some older literature some high-valence metal oxides are incorrectly named peroxides even though they do not contain the peroxide ion. Defect formation in fluorite dioxides. Both CEO2 and O2 accommodate excess oxygen in their lattice by forming peroxide molecules. This is in contrast to UO2, which takes in excess oxygen by forming charged interstitials. The accommodation of excess oxygen in these systems changes their thermal and diffusion properties. This will have implications in the oxide's use in nuclear fuels and solid oxide fuel cells. Adding oxygen to the CEO2 system, for example, will increase the diffusivity of the oxygen, allowing for more efficient solid oxide fuel cells. Peroxide salts bonding in O22-. The peroxide ion is composed of two oxygen atoms that are linked by a single bond. The molecular orbital diagram of the peroxide dianion predicts a doubly occupied anti-bonding pi* orbital and a bond order of 1. The bond length is 149 pm, which is larger than in the ground state of the oxygen molecule. This translates into the smaller force constant of the bond and the lower frequency of the molecular vibration. The peroxide ion can be compared with other molecular oxygen ions, superoxide O2- and ozonide O3-, but contrary to them, The peroxide is not a radical and not paramagnetic. Owing to the weak bonding between the oxygen atoms, peroxide easily undergoes homolytic cleavage, yielding two highly reactive radicals. This cleavage is accelerated by temperature, illumination, or chemical reactions. Preparation of peroxide salts. Most alkali metal peroxides can be synthesized directly by oxygenation of the elements. Lithium peroxide is formed upon treating lithium hydroxide with hydrogen peroxide. 2LiOH plus H2O2 Li2O2 plus 2H2O. Historically, barium peroxide is prepared by oxygenation of barium oxide at elevated temperature and pressure. 2Bau plus O2 2Bau. 2 barium peroxide was once used to produce pure oxygen from air. This process relies on the temperature-dependent chemical balance between barium oxide and peroxide. The reaction of barium oxide with air at 500 degrees Celsius results in barium peroxide, which upon heating to above 700 degrees Celsius in oxygen decomposes back to barium oxide, releasing pure oxygen. Properties of peroxide salts. Few reactions are generally formulated for peroxide salt. In excess of dilute acids or water they release hydrogen peroxide. Upon heating, the reaction with water leads to the release of oxygen. Instead, the peroxide anion is a stronger nucleophile than hydroxide and displaces hydroxyl from oxyanions, e.g., forming perborates and percarbonates. Sodium perborate and sodium percarbonate are important consumer and industrial bleaching agents. They stabilize hydrogen peroxide and limit side reactions. The peroxide anion forms an adduct with urea, hydrogen peroxide, urea. Hydrogen peroxide is both an oxidizing agent and reducing agent. The oxidation of hydrogen peroxide by sodium hypochlorite yields singlet oxygen. The net reaction of a ferric ion with hydrogen peroxide is a ferrous ion and oxygen. This proceeds via single electron oxidation and hydroxyl radicals. This is used in some organic chemistry oxidations, e.g., in the Fenton's reagent. Only catalytic quantities of ion is needed since peroxide also oxidizes Fe to ferric ion. The net reaction of hydrogen peroxide and permanganate or manganese dioxide is manganese ion. However, until the peroxide is spent, some manganese ions are reoxidized to make the reaction catalytic. This forms the basis for common monopropellant rockets. As a ligand in coordination chemistry, peroxide functions as a bidentate ligand in a variety of coordination complexes. Some complexes have only peroxide ligands, e.g., chromium oxide peroxide 42-. Similarly, molybdate reacts in alkaline media with peroxide to form red peroxymolybdate Mo4 2-. The reaction of hydrogen peroxide with aqueous titanium gives a brightly colored peroxy complex that is a useful test for titanium as well as hydrogen peroxide. Many transition metal dioxygen complexes are best described as adducts of peroxide. Applications. Many inorganic peroxides are used for bleaching textiles and paper and as a bleaching additive to detergents and cleaning products. The increasing environmental concerns resulted in the preference of peroxides over chlorine-based compounds and a sharp increase in the peroxide production. The past use of perborates as additives to detergents and cleaning products has been largely replaced by percarbonates in order to decrease the emission of boron to the environment. Sodium percarbonate is used in such products as OxiClean and Tide laundry detergent. When dissolved in water, it releases hydrogen peroxide and soda ash. 2Na2CO3 1.5 H2O2 2Na2CO3 plus 3 H2O2. The use of peroxide compounds in detergents is often reflected in the trade names. For example, Persil is a combination of the words perborate and silicate. Some peroxide salts release oxygen upon reaction with carbon dioxide. This reaction is used in generation of oxygen from exhaled carbon dioxide on submarines and spaceships. Sodium or lithium peroxides are preferred in space applications because of their lower molar mass and therefore higher oxygen yield per unit weight. Alkali metal peroxides can be used for the synthesis of organic peroxides. One example is the conversion of benzoyl chloride with sodium peroxide to dibenzoyl peroxide. Organic peroxides. Organic peroxides can be divided into two major classes, peroxyacids and organic hydroperoxides. The first class is derived from the carboxylic acid and the second from ethers or alcohols. Preparation. Most peroxyacids can be obtained by the reaction of hydrogen peroxide with the corresponding carboxylic acid. R equals organic group. Another route employs acyl halides instead of the carboxylic acid. This method is used primarily with aromatic compounds under basic conditions in order to neutralize the resulting hydrogen chloride. Aromatic aldehydes can be auto-oxidized into peroxycarboxylic acids. R equals aryl. The products, however, react with the initial aldehyde forming the carboxylic acid. Several synthesis routes are known for aliphatic peroxides, such as the reaction of dialkyl sulfates with alkaline hydrogen peroxide solution. In this method, the alkyl sulfate donates the alkyl group and the sulfate ion forms the leaving group. This method can also yield cyclic peroxides. The four-member dioxetanes can be obtained by 2+2 cycloaddition of oxygen to alkenes. He-selective synthesis of hydroperoxides can be carried out by free radical oxidation of alkanes with oxygen. Here the active site formed by a radical initiator reacts with oxygen to form a hydroperoxyl. The addition of oxygen results in a more active radical which can further extract hydrogen atoms and release the hydroperoxide, leaving a new radical. This process is used industrially for the synthesis of phenol from benzene and is called the Cumena process or Hock process for its cumena and cumena hydroperoxide intermediates. This auto-oxidation reaction can be used with common solvents from the group of ethers, such as diethyl ether, diisopropyl ether, tetrahydrofuran or 1,4-dioxane. It yields a volatile hydroperoxide ether that upon heating can result in a serious explosion. Peroxides are formed by living organisms through ene reactions or Diels-Alder reactions between alkenes and oxygen. Unsaturated fatty acids can serve as the olefinic substrates for the ene reaction, and unsaturated amino acids like histidine can be the reactant for the Diels-Alder cyclization. Rancidification of fats is partly caused by the formation of peroxides. Properties and applications of organic peroxides. Peroxycarboxylic acids are generally weaker acids than the parent carboxylic acids. Like most peroxides, they are strong oxidants and tend to explode at high concentrations and higher temperatures. Organic peracids are used in the synthesis of epoxies via the Prielgev reaction. Another important application is the synthesis of lactones or cyclic ketones in the Beria-Villager oxidation process. In both cases, electron-poor peroxycarboxylic acids are especially efficient, such as metachloroperoxybenzoic acid. Tert-butyl hydroperoxide is a common oxidant in the Sharpless epoxidation, which is used for the stereoselective synthesis of epoxides. Carl Barry Sharpless was awarded the 2001 Nobel Prize in Chemistry for this reaction. Peracetic acid is a popular disinfectant in the medical field and food industry. Various peroxide solutions are commercially produced for the cleaning and disinfection of contact lenses. Dibenzoyl peroxide is used as a radical initiator. Its sweet peroxide bond cleaves easily, yielding reactive benzoxy radicals, which assist polymerization leading to plastics like polyethylene. One of the synthesis methods of the commercially important plastic caprolactane, the precursor to nylon 6, is a Beria-Villager rearrangement of cyclohexanone with peracetic acid. This yields caprolactone, which is then converted to caprolactane by reacting it with ammonia. Industrial resins based on acrylic or methacrylic acid esters are invariably produced by radical polymerization with organic peroxides at elevated temperatures. The polymerization rate is adjusted by suitable choice of temperature and type of peroxide. Some peroxides are drugs, whose action is based on the formation of radicals at desired locations in the organism. Artesunate is also efficient in reducing egg production in schistosoma hematobium infection. Many organic peroxides can initiate explosive polymerization in materials with unsaturated chemical bonds, and specifically triacetone triperoxide and hexamethylene triperoxide diamine are powerful explosives. TATP is an inexpensive compound and is relatively easy to make, whereas most other potent explosives, such as trinitrotoluene or RDX, contain nitrogen, which is relatively easy to trace by sniffing techniques. TATP is nitrogen-free and therefore is very difficult to detect by conventional screening methods. For this reason, it is an explosive favored by terrorists. TATP and HMTD were used in several executed or planned terrorist acts of the early 2000s, most notably in the 2001 shoe bomb plot and the 2005 London Underground bombings. Several detection devices have been designed since those events. One, for example, releases a chemical mixture which changes color when interacting with traces of TATP. Laboratory identification. Several analytical methods are used for qualitative and quantitative determination of peroxides. A simple qualitative detection of peroxides is carried out with the iodine-starch reaction. Here peroxides, hydroperoxides or peracids oxidize the added potassium iodide into iodine, which reacts with starch, producing a deep blue color. Commercial paper indicators using this reaction are available. This method is also suitable for quantitative evaluation, but it cannot distinguish between different types of peroxide compounds. Discoloration of various indigo dyes in presence of peroxides is used instead for this purpose. For example, the loss of blue color in leucomethylene blue is selective for hydrogen peroxide. Quantitative analysis of hydroperoxides is performed using potentiometric titration with lithium aluminium hydride. Another way to evaluate the content of peracids and peroxides is the volumetric titration with alkoxides such as sodium ethoxide.